A team of Stony Brook University researchers is working on computer models that could help speed the discovery of drugs to combat the novel coronavirus responsible for COVID 19. The researchers are working on models to better understand how the spike protein on the surface of the COVID 19 virus interacts with the cell it infects. These spikes all around the exterior of the spherical virus particles give the virus the look of a crown and its name as a coronavirus. They are also the part of the virus that attaches to a cell inside people's respiratory systems using a specific protein on the human cell to begin the process of causing infection. Because of this role, the spike protein is also the main target for antibodies that would provide immunity to coronavirus and for developing drugs that potentially could block infection. The researchers are using computer models to understand more about how the spike works. Scientists have already captured useful pictures of the spike with atomic level detail, and they have a good idea of what the spike is doing. For example, they know that the spike changes its shape as it contacts the cell, and this leads to the virus and cell merging. But they need to know more detail in order to control it. For example, you may know that your car door is the way you can get in and out of your car. But if you have only seen the door in a picture and have never watched someone actually open a door, you don't really know how it works. You also probably would not guess that the handle is the part that you need to move to get the door to open, and that perhaps there is a key that can lock it and keep it from opening. The researchers are working to understand the process at more detailed level. For example, what exactly leads to the spike of the virus opening the door to infection when it contacts the cell? And are there places on the spike away from this contact point that could act like keys, where scientists might be able to make a small molecule that would bind to it and lock it closed? The researchers will be looking at 60 different target sites where a new drug may attach to the virus and 1 billion drug-like molecules that might be used to modify the door handles, hinges or other components so we can identify the most promising options for neutralizing the virus to keep it from entering cells. Soon the computer models will be scaled up to run on supercomputers. It will help sort through the details of these virus cell interactions to help unlock an understanding of the COVID 19 virus spike protein.